talk. And Martina, I'd ask you to take it away, please.
Thanks, David. So today we have the pleasure of welcoming Dr. Russell Morton, an assistant professor from the Department of Neurosciences at the University of New Mexico Health Science Center. Dr. Morton completed his Bachelor of Arts degree at the University of Colorado in 2003 in molecular, cellular, and developmental biology. He then completed his PhD in 2011 at George Washington University that cooperated with an NIH graduate partnership program. And then in 2016, he completed his postdoctoral fellowship at the University of New Mexico School of Medicine. He currently has a grant from NIGMS Center for Brain Recovery and Repair. His research group is primarily focused on understanding the complex set of events that occur immediately following mild traumatic brain injuries and how those events can have long-term impacts on cognitive function. He's also interested in how these early events are exacerbated in repeated injuries. Dr. Morton's laboratory is currently investigating the following questions. Are spreading depolarizations a key underlying mechanism in acute behavior and post-traumatic headaches following mTBIs? Is the threshold for spreading depolarizations altered by a previous mTBI? What is the role of spreading depolarizations in the network dysfunction of mTBI? Their long-term goal is to identify key early events of mTBIs that can be targeted to help promote brain recovery and repair. In addition to his research, he is a passionate educator and mentor, strong believer in science communication, and has participated and facilitated multiple Alan Alda Center for Communicating Science workshops. Now over to you, Dr. Morton.
Hey, thank you very much, Martina. Thank you very much for the introductions. I want to also extend a thank you to Josh, Adam, and David for creating this platform for us to all communicate via virtually, being these difficult times that we're in. So thank you all for joining. I first want to just give a real big shout out to the Center for Brain Recovery and Repair here in New Mexico. But none of this really would have been possible without that. And also the preclinical core, which is part of that Center for Brain Recovery and Repair. Of course, the Department of Neurosciences as well. So as Martina mentioned, we're really interested in what role that these spreading depolarizations might play in mild traumatic brain injuries. So with that said, I just want to give a brief outline because I want to give a very brief introduction. mTBIs do not really need too much of an introduction for this group. But then I want to talk about SDs and mild traumatic brain injuries and how they may contribute to the acute behavior. And then hopefully there's time, I'm going to briefly talk about some preclinical cognitive deficits following repeated injuries and how we may use that as a preclinical model for repeated injuries. So obviously this is my very brief introduction. Mild traumatic brain injuries are a very serious concern, but I think not only the highlight often focuses in on sports-related concussions, which are definitely a problem, but also due to falls and accidents, and particularly motor vehicle accidents, contribute to a significant amount of these mild traumatic brain injuries. So we're all very familiar with the idea that there's approximately 2 million mild traumatic brain injuries per year in the US. And I often like to think of things in a more physical manner. And if we were to convert that to number of gallons of water, we could basically fill 3 Olympic-sized swimming pools every year with mild traumatic brain injuries. And when you think about it in that kind of a scale, it's a real big problem. And I think that there's a lot that we don't fully understand regarding these injuries. And it's something that needs constant investigation, which all of you are very familiar with. I also want to put up the consensus statement from one of the international conferences on concussion in Berlin that happened in 2016. This is from the publication that came out of that. But there's 3 things that I wanted to highlight with this definition. I like this definition for multiple reasons, but some that are important for today's talk is one of the first ones is this impairment of neurological function that resolves spontaneously. And we'll see why you think why I think that's important in these injuries, getting into my talk, but also the clinical signs and the symptoms that are often talked about and looked for recovery are largely a reflection of the functional disturbance rather than some structural damage, which I think is also one of the key aspects of these injuries. And one of the other things is that these injuries may or may not resolve in the loss of consciousness. And I think that's a real important thing that often goes unlooked or overlooked, especially in the public, is that they often think that if there's no loss of consciousness, then there really was no injury. And I think probably everyone on this call contributes to the understanding that that's not the case and that these are serious injuries. So one of the things I was most intrigued about was what really happens to the brain during a concussion. So what are the very first kind of events that happen? So to do that, we often adopt rodent models of TBI models. And there's a lot of different models, and there's variations among all of these. And as Dr. Doug Smith once told me, a TBI researcher would rather share a toothbrush than share a TBI model, right? We all have our favorite way of doing things. And so this is a nice paper by Sham et al. In 2013, highlighting some of these preclinical models that involve everything from fluid percussion injury and CCI, controlled cortical impact, to more simple weight drop models, which often are closed skull injury models. And through all of these, a combination of all of these different models, there has been a lot known over the last, say, 20, 30 years involving what happens in the brain in those first few minutes after an injury. And so one of the first to kind of describe these early events was actually Takahashi in the early 80s, used a fluid percussion injury, which is a fairly mild injury, but then looked at two things, one being the extracellular potassium, and looked at brain activity with this EEG. And what he noticed was that in the first minute or so, you see this kind of suppression of this EEG high frequency activity. And this also correlated with a very, very high increase in extracellular potassium. About a decade later, Katayama and others again showed this very prominent increase in extracellular potassium, but it also showed that that was also associated with this dramatic increase in extracellular glutamate, and also to a lesser extent serine as well. About the same time, Nielsen and others were using some dialysis studies to look at other things. And what they found was, in the first few minutes following a mild injury, there was these very prominent increases in adenosine and lactate. And again, about that same time, autoradiography studies by Yoshina and others showed that there was a dramatic increase in the glutamate uptake in the first few minutes of the ipsilateral cortex relative to the contralateral cortex, that then decreased over the first, say, 6 hours, and then remained relatively low for multiple days. And so one slight caveat to some of these initial studies was that even though some of these injuries were very mild, the combination of controlled cortical impact and fluid percussion still requires some surgical procedures to be done prior to the injury, such as burr holes or craniotomies and things like that. So there always is that potential of prior injury prior to the actual mTBI. Now, all this has really been nicely conceptualized by Giza and Havda back in 2014 in their paper that basically described the culmination of all of these studies as a neurometabolic cascade, and that you see these huge increases in extracellular potassium and glucose and extracellular glutamate, this huge increase in intracellular calcium. But what I haven't shown, but others have, is that there's this prolonged period of reduced cerebral blood flow and reduced glucose uptake. Anguissa and Havdas and others have shown that if you have injuries during this period, that the injuries have worse outcomes and are associated with more damage. And so, you know, collectively, we kind of assume that it's these early days that seem to be a sensitive period or vulnerable period for the brain for secondary injuries. I think this is kind of formed the foundation of you know, this early rest and removal of not activity, but from sports related play and things like that for the first few days. And so this is not a new concept by any means, but this has always kind of been proposed to be initiated by spreading depolarizations or spreading depressions. And so what are spreading depolarizations or spreading depressions? Well, these have been around for a very long time. They were first described by Liao in 1944 in this kind of seminal paper where he was using serial electrodes spread across the cortex of rabbits. And then in the very rostral part of the brain, he would use an electrical stimulus. And if that electrical stimulus was sufficient enough, what he found was that beginning with some of the first channels, the channels of this high frequency activity of the brain would then suppress and it would get very quiet. And that suppression would then propagate through all of the electrodes, eventually the entire cortex being quiet. And then what he found was that it spontaneously revolved in that same order, where the first to resolve were channels 1 and 2, eventually going back to full activity across the entire cortex. Now, one of the things that I think are important in relationship to my talk today is that even in this initial description of spreading depressions, Liao mentioned that these events could be initiated with a mechanical stimulation. So simply just touching the brain with a small glass rod could easily evoke these events. And so as Liao initially described these as spreading depressions because of this depression of activity, we've actually learned that that suppression is associated with a huge depolarization of the entire cortical tissue, so including astrocytes and neurons. That suppression is then extended by a depolarization block and adenosine and things like that to suppress activity. And once that ion gradients kind of recover and things can then go back to normal, that high frequency activity comes back. And so years of research looking at this now kind of uses the term spreading depolarization instead of depression because we know that there's a lot more going on. But these are massive events that significantly alter brain physiology. And so sometimes they're often referred to as brain tsunamis because they're such large events. So here I show you these events are so large that they can actually be seen by the naked eye. So this is what we typically use as intrinsic optical signal imaging. And so what we're doing here is just getting a camera, looking at the surface of the cortex, and just using white light and no filters at all, we can initiate one of these events here in the lower left-hand corner and see these basically with our naked eye. And so as you can see, these events propagate through the cortical surface. The reason that these, even though we can see them with our negative eye, they usually go undetected is because they propagate at a very, very slow rate. So anywhere between 3 to 7 millimeters per minute. And so this video here is sped up actually 4 times the real time. And so when they propagate so slow, it's very hard to see, but it is possible. So what all goes on during one of these events? Well, as I've kind of shown you before, that there's this huge depolarization of the cells. And so that is associated with a huge drop in the extracellular voltage as all the cells depolarize. That is also associated with this huge increase in extracellular glutamate and in extracellular potassium. During this time, there's a huge increase in intracellular sodium and intracellular calcium as well. So I hope that all of this looks very similar to everyone, because I've just showed you basically the same thing occurring here. And that's why it's often been proposed that SDs kind of initiate this neurometabolic cascade, is because all of these events line up in time with the initial injury. So SDs were long thought to be a rodent phenomenon and not really associated with human physiology. And that was all changed in the early 2000s, where Strong and others actually showed that in moderate and severe TBIs, these events happen. And the reason that they were able to detect these events more readily is because the injuries were so severe that the craniotomies needed to be done to relieve intracranial pressure. And when they were able to do that, as shown in this figure here, is that once they did the craniotomy, remove part of the skull, they could actually slide in electrodes to be directly on the surface of the brain. Because these events move so slow, they're often filtered out by the skull and other recording artifacts that are typically done in normal EEG, it's hard to detect these events with just typical EEG. So what they showed is that early on, so this is about approximately 29 hours after the injury, they could find these propagating events of depression, which is associated with the spreading depolarization. What they also noted was that as the injury kind of progressed here, and as you can kind of follow along in this graph, that as the injury got larger and larger, this is at 38 hours, the depressions became more frequent, and that depression of activity actually got longer. And so what's really been focused on in the last, say, 10 or 15 years is this idea that SDs, or spreading depolarizations, propagating through healthy brain, as shown kind of by this electrode D, that depression is relatively small. The tissue can easily recover. But what happens is when the spreading depolarization actually travels through compromised brain, so brain that has inadequate blood supply, the spreading depolarization and the depression occur, but that tissue is compromised in some way where it can't fully recover. And that tissue then goes on and dies, and this leads to injury progression. But this really has left the door open to, do SDs occur in more mild injuries where we don't have to do a craniotomy? And so this type of mild model was really highlighted very well by Dr. Robel last week. And there's certain features that we thought were very important to more accurately model a concussion. And one had to be the closed skull. And we didn't want to induce any injury prior to the induction of the mTBI. One thing, going back to the consensus statement, was that we really wanted a model that lacked physical damage, right? When there's large tissue damage, the SDs are going to be initiated just because of the damage, not necessarily the injury. And one thing that we thought was important was to unrestrict the head, and that the head was allowed to move and respond to the impact. And so what we kind of decided on was a model that's been used previously, but we used the cortical impactor as other studies have, but instead we just left the skull intact, the skin intact, and we removed the head restriction. So the animal's head lies in this chiasm foam platform that keeps the head in orientation, but doesn't restrict its movement at all. The diameter of the tip is about 5 millimeters. The velocity of that tip traveling down is about 4 meters per second, with a deflection of 5 millimeters. And so these studies were initially done by a very motivated and talented undergraduate in my lab, Ashley Hans-Lanham. And so we wanted to center this impact over the midline, roughly over the medial prefrontal cortex, which is about approximately 1 millimeter from bregma. However, since we leave the skin intact. We don't really have an anatomical way to measure. We typically use the eyes as a way of orienting the impactor to the skull. I must say that all of our impacts are done under anesthesia. Most often they're done under 2% isoflurane. In some cases where the animal is maintained under anesthesia, we usually use urethane. But we use animals that are basically young adults, 8 to 12 weeks of age. And all of our experiments include equal numbers of both male and females. And in some cases, we do see differences between the two, and we'll talk about some of those. So one of the things that we noticed very early on, actually the first time we did these studies, was that in the sham-treated animals, which is up here in the upper left, once the animals were placed back into a home cage to recover from anesthesia, they typically recovered very quickly and started to roam around. Whereas when we did the mTBI treatment, the animals remained motionless for a very long period of time. And I was actually really encouraged to hear that Dr. Robel last week actually sees a very similar behavioral phenotype. But if we quantify this, we see that the mTBI animals have a significantly longer latency to write, and also a latency to movement as well. And we felt like this was a pretty accurate model of a concussion-like injury, right? We wanted to quantify this a little bit more, and so we actually kind of repeated these studies in an open field arena. So the animals were under anesthesia, they received the mTBI or sham injury, and the animals were placed into the center of this open field arena. We use a motion tracking system for 10 minutes to then monitor the animal's recovery and then their movement after they recover. And what these heat maps kind of show is quantified here, and that the mTBI animals remain motionless, like I just showed you in the previous video, in the center of the arena. But once they recover, they typically find a corner and then just kind of hunker down and don't explore much after that. And so this is quantified here, showing that the mTBI animals showed a reduction in total distance traveled, an increased latency to actually reach the border. Most animals prefer to be in the borders and not in the center, so it's a motivation to then move, to get themselves out of the center and move towards the border. And then after they recovered, they had this severe reduction in episodes of movement after their recovery. We actually extended this a little bit and we did some home cage monitoring, and where the animals were singly housed in this photo beam activated activity system by San Diego Instruments. And so then we acclimated the animals to these behavioral setups for 24 hours. And here, what I'm showing you here is the cumulative total beam breaks during the dark cycle, so during their most active cycle. Here, in their previous cycle, their first day in the cage, what we see is that there's no statistical difference between the sham and mTBI animals. Not surprising, because they've not gone under any experimental condition. However, 2 hours after the beginning of their dark cycle stage, they received a sham treatment or mTBI, and then they were monitored for the following 6 hours. And what we find is that even upon recovery and back in their home cage, the mTBI animals still have an overall reduced home cage activity for the roughly 4 hours after the injury. Again, as I mentioned before, we looked at just gross tissue damage. We used a Nissel's stain. I'm showing representative male and female images here, but we don't really see any gross tissue damage in the cortex, and we don't see any major bleeding or anything like that. We also looked for individual cell death. So we used the Fluorojade stain, which I'm sure most people are familiar with, but we were really unable to detect any significant cell death following the injury. We looked at day 1 and at day 10, and we were unable to find significant differences. And this is just quantified kind of gross fluorescence, but we didn't see any individual cell bodies seem to be lighting up. We also looked at the common marker GFAP to look at astrocyte activation. Again, we looked at day 1 and day 10, and we didn't really find a significant increase in GFAP expression in the cortex or shown here in the corpus callosum. So we can definitely more readily detect them in the corpus callosum, but we didn't see any increases in that fluorescence. We also went through and looked pretty significantly for blood-brain barrier disruption. To do that, we used this Persian blue stain, and we were able to find a few cases of blood-brain barrier disruption. We found these examples in 2 of the 8 animals that had the mTBIs. While one of these bleeds was in the cortex, which is just outside of the impact zone, we did find evidence of this other one that was non-cortical. So we don't think that these play a major role in the injuries, given the very few animals that we detected this in, and these were really the only 2 cases. We did find one animal that did have increased tunnel staining, and this was in the cortex, but again, it was only in one animal, and we didn't find any other cases in the other animals. So we don't think there's a whole lot of cell death following these injuries, and this was done at 24 hours as well. We also did a bunch of behavioral assays, and I don't want to bore everyone more than I already have by showing all this data, because we did a lot of this at 24 hours. We did open field and novel object recognition, accelerating rotor rod, conditional trace sphere, and we really didn't find any significant differences. And this is all published in our recent paper in eNeuro. We did find some small differences in gait, but they weren't very large differences. Again, I'm not fully confident on how significant those small differences were. But we were really interested in this very, very acute behavior. There was something clearly going on during these early events. And so to extrapolate this a little bit more, my current graduate student, Natalie Pinkowski, has worked diligently to establish this series of behavioral tasks that other people have used as well, that are collectively scored as a neurological severity score. And so that includes our normal open field, as I've shown you before. It then includes an escape and exploratory activity, again, gross gait analysis, startle response, forceps grasp, inverted mesh, and then elevated beam walk with reducing diameter beams, and concludes with a 1 centimeter round beam. And so these are basically scored on a 0 to 1 pass or fail scale, with 11 being the worst. And so as we all kind of worry about in science, we worry that once we change something or put another cohort, things might change. But indeed, that was not the case. In this cohort of animals, again, we saw the increased latency to right themselves, and also this increased latency to movement, which is comparable to what we saw before. But then Natalie was able to show that there is also this associated increase in the neurological severity score with the TBI relative to the sham treated animals. But again, as I keep mentioning, we're really interested in what was happening in the brain. And so we have this nice behavioral phenotype that we've established, but we really want to know what was going on in the brain. And so the first stab at this was done actually by a talented post-bac student, Johan Pacheco, who's now in medical school. And what we did was we paired our mTBI injury with laser speckle contrast imaging. So for those of you who may not be familiar with this, this is an imaging technique that can image cerebral blood flow basically through the skull. And so what we would do is we would generate a skin flap, and so we could expose the skull for imaging, but have the skin present for the impact. The impactor was placed right behind the imaging camera. Immediately following the impact, the animal was then moved back under the camera to look at the cerebral blood flow responses. And so with that, what Johan saw was that once we pulled the animal back underneath the camera, we saw this propagating wave of hypo cerebral blood flow. This is associated with spreading depolarizations. And so if the video is not working here, we have still images as well showing that these events then propagate through both hemispheres because our injury is over the midline, we do typically see spreading depolarizations in both hemispheres. And then if we look at specific regions of interest, what we can see is that this propagating wave of hypoperfusion is seen immediately after the impact. Johan then extended this to look at not only this propagating wave, but what happens kind of not long term, but in the first few minutes. And what Johan found was that after that propagating event very early on, in the next roughly 90 minutes, there's a long period of reduced cerebral blood flow, both in the tissue and in the venous structures. That reduces about 30%. And then, as I mentioned, fully recovers around 90 minutes. To kind of look more chronically at this, to see if that after that 90 minutes, if the cerebral blood flow then is stable for the next few days, Carissa Milliken and her dog here established a nice non-invasive cranial window where basically it includes a small incision into the skin that's then sealed with cyanoacrylate glue that can be left in for multiple days or months even, and still allows for laser speckle imaging. And so what Carissa was able to show here was that in sham treated animals, that cerebral blood flow maintained relatively the same over that 30-minute period and also for multiple days. Carissa was also able to show that, again, with our mTBI model, there's that dramatic decrease in the cerebral blood flow at 30 minutes that then recovered and remained relatively stable for the few days after. So we think that at least at 24 hours, things physiologically seem to be back to normal. But we'll talk more about that a little bit later. So about the same time that we were looking at this propagating wave of hypoperfusion, another group, Niles Henninger's group, put out a paper showing just that they had a weight drop model that showed this dramatic decrease in cerebral blood flow that lasted, in their hands, they went out to 60 minutes. And what they were able to show was that the injuries with lower weight didn't induce this type of changes in cerebral blood flow. And they went on to show that, again, just as I just showed you, that these events are associated with the cortical spreading depression, as they show here. And what they showed was that in the animals that had the SD, that then was followed by a reduction in the cerebral blood flow. And the animals that did not have an SD did not have a significant reduction in that cerebral blood flow. And just as I've shown you previously, those injuries were then again associated with an increase in this neurological severity score, a very similar measure as I just showed you. And again, they showed here, using that intrinsic optical signaling that I mentioned earlier, that indeed these are propagating events and they are typical of SDs. But we were really interested in, could we actually detect the electrical physiological responses of the SDs? And so what Johan did in combination with the laser speckle imaging that he was doing was that we were able to introduce a small burr hole into the very caudal part of the brain through the skull. And then once the animal had the treatment, he would pull the animal back underneath the camera and carefully insert a glass electrode into the cortical surface. So if we did these in sham treated animals, what we saw is we record a series of baselines for a few minutes. Then we remove the electrode for the actual impact. Then we place the electrode back into the cortical surface. And what we saw was that the DC recording remained stable. The high frequency activity remained stable. And this is just a high frequency power, the power of that high frequency. However, this was much different in our mTBI model. What Johan was able to show was that indeed these propagating waves of the hemodynamic responses, which I just showed you, are associated with DC shifts, so that extracellular drop in potential. and also with that suppression of high frequency cortical activity that again recovers spontaneously within about 8 minutes or so. So indeed, these are classical spreading depolarizations. Another very talented undergraduate student in my lab also kind of repeated what the Bally and others showed, was if we reduced our impact speed, so in this case, we used 2 meters per second, what Claire was able to show was that if we have this reduced speed, we actually don't get that, we don't initiate an SD. And so here what we show is individual images, but here's the raw trace where we don't see any propagating waves of hyperperfusion. We also showed that this reduced speed of 2 meters per second did not have the behavioral phenotype as our normal mTBI model does. And here we show no significant difference in latency to movement and to the writing themselves as well. Another talented undergraduate student, Katie McCurdy, she actually asked the question of, could we block these events using typical blockers of spreading depolarizations? And so, as you can imagine, since these are such large events, the blockers that do seem to work, ketamine being one of them, we have to use basically anesthetic doses of these agents to completely block the SD. But she was able to show that in saline-treated animals, again, we have this propagating wave of hypoperfusion, as shown here in the red trace. And then with a ketamine dose of 120 milligrams per kilogram, we could effectively block the spreading depolarization. She showed that there's a significant reduction in the number of animals that have SDs with ketamine on board. So 2 of the 7 animals did have an SD, even though ketamine was there. What we did after that was look at the animals that did not have an SD associated with the mTBI. We then looked at the cerebral blood flow 30 minutes post. So typically, as seen here in the saline, we have that about 30% reduction in cerebral blood flow that was not present if the ketamine was on board and it blocked the SD. We do have a few animals here that seem to increase their cerebral blood flow, and we're not exactly sure if that's an effect of anesthesia or the combined effect of urethane and ketamine. We're still trying to parse that out, but at least we don't see any reduction in cerebral blood flow. So next, we wanted to kind of, as I mentioned before, the ketamine experiment, to actually block the events, they're anesthetic doses, so it doesn't really allow us to do much behavior. And so what we wanted to do was come up with a way that we could initiate spreading depolarization in the complete absence of any injury or mTBI. And so my graduate student, Natalie Pankowski, has started doing this, where she implants bi-hemisphere cannulas. And so 1 cannula into each hemisphere. KCL is a known activator of spreading depolarizations, and is commonly used within the field. And so what she has been able to show is that if she uses microinjections of 0.75 microliters of 1 molar KCL, she can reliably induce these spreading depolarizations. So this is just the intrinsic optical signaling gathered from this series of images. Next, what Natalie was able to do was actually induce these spreading depolarizations in the absence of any impact, and then do our typical neurological severity score. And what Natalie was able to show was that indeed the SDs alone were able to were associated with this increase in the neurological severity score. And so this was actually very similar to our mTBI data that I showed you earlier. So this shift in the neurological severity score. One caveat to the cannulas that I just showed you is that, as I mentioned in the very beginning, we typically do not want to have any injury associated prior to our injury or our mTBI model. And so the introduction of the cannulas, while very, very useful for inducing SDs, we still have to implant those cannulas prior to the mTBI model. And so recently we've also adopted an optogenetic approach, in where the channel rhodopsin is expressed under the Thi1 promoter, so most glutamatergic neurons. If we then flash the cortex with light, so this is through the skull, you can see this propagating wave, which is the SD going by. If we graphically look at that trace, it's shown here, so you can see the propagation and the changes in the intrinsic optical signaling. So now we feel like we have a nice non-invasive model to induce SDs in separation of the impact, to identify the role of SDs specifically. Natalie's also been able to recently adopt our laser speckle contrast imaging, which I showed you earlier from that Johan had done, but do this in awake and moving animals. And so here, Natalie has been able to do some head restriction. So the animals are placed within this head restriction, but allowed to move freely on these styrofoam balls. And so Natalie just showed here that indeed, if the animals receive an impact, then she puts them on this apparatus, we can detect the propagating wave of hypoperfusion as associated with the SDs. And so now we feel like we have a good way of non-invasively imaging, and also allowing the animal to be freely moving while imaging. So to kind of wrap up this first part a little bit, I want to leave plenty of time for discussion, but what I've shown you is that we do have this acute behavior of reduced mobility early on. That's clearly associated with the spreading depolarizations. This is also associated with an increase in the neurological severity score, and that we can specifically induce SDs in the absence of an impact and result in similar neurological severity score deficits. To kind of switch gears a little bit, we have we also, obviously this is in rodents, right? And so we don't know, and I'm not sure, if we will ever know if they happen in humans. But I wanted to highlight some of this data from David Carmelo's lab from Stanford, where they use accelerometers placed in the mouth guard of their football players. And so this was published a few years ago, but they were actually able to record these events of head acceleration from a player who did receive a full-blown concussion. And what's just shown here is a different axial accelerations and rotational acceleration of those sensors. And so with this, they were actually able to model what might be happening in the brain. So what's shown here is first principle strain. And so the warmer colors indicate more strain, and they kind of modeled what might happen with the accelerations that they recorded from the actual mouth guard. And so if you are familiar at all with SDs, seeing this amount of shear stress and overall movement of the brain, I would be surprised if there's not SDs initiated in an event like this, if there's that much movement and shear stress. So we really think that the spreading depolarizations are a key underlying mechanism of some of this acute behavior. And we think that, obviously, as has been suggested before, many of these initial events are most likely caused by a spreading depolarization. But we're really interested in what role these spreading depolarizations may have in this more short-term window, right? For the first few days, are SDs really contributing to this overall reduced cerebral blood flow or reduced glucose uptake? And that's kind of stuff that we're focusing in on in the future. In light of that, I just want to talk about how SDs how do they may contribute to some of these deficits. And so SDs are kind of a vicious cycle if you're not familiar with these. So once an SD occurs, the tissue undergoes a lot of energy expenditure to reestablish those ion gradients. And so as you can imagine, this takes lots of energy. You use up a lot of ATP. In the wake of an SD, there's an uncoupling between the neurovascular unit. And so the vascular becomes unresponsive to this increase in need. And And so you get a reduction in glucose and a reduction in oxygen, which both contribute to the initiation of another SD. And so this is done in a pig model, a stroke model, but what you can see is that there's these propagating waves that just keep reinitiating from certain regions. And so it's part of this vicious cycle where more SDs just seem to occur. And when the tissue is then compromised, that's when you can get cell death and things like that. So we think that this may be a really important thing to understand in the longer term, more like in the days and perhaps even months after. So last thing I want to switch to very briefly is we also are very interested in some of these more long-term cognitive deficits. And so one way that we've tried to do this is how can we really establish a good cognitive measure in the preclinical model. And so to do that, we've established this touchscreen-based visual discrimination task. This was done by our preclinical preclinical core technician, Haiku Zhang, that used this touchscreen-based system where one of the visual stimuli were paired with reward and the other was paired with a timeout. And if we use this task, we first did a single impact, and that impact was done 2 days prior to the very beginning of this touchscreen learning. And what we showed was that the single injury, the animals performed very, very similar to the sham treated animals. So they had similar correct trials, incorrect trials, and correction trials. So correction trials are if they respond incorrectly, the two images are shown to them in this very same orientation repeatedly as a correction trial until they respond correctly. And then we have a criteria of once the animals perform at 85% correct, then the animals go on or complete the task. And so they had a similar number of total discrimination trials. Once they achieve that 80% criteria, the animals are then reversed. So the previously rewarded stimuli is now incorrect and the other one is correct. And so the animals must unlearn the previous association and relearn the new association. Again, the single injury animals did not behave any different than the sham treated animals. And so months of behavioral testing went by and we really had nothing to show. And we were kind of disturbed by this and depressed. But when we sat back and thought about it, it wasn't really surprising, right? We typically don't see cognitive deficits following a single injury. It's really this multiple impacts, repeated injuries that seem to be associated with the more long-term cognitive deficits. And so to look at that more directly, we did 4 impacts spread out 24 hours apart, so 1 every 4 days. Animals were allowed to recover for 2 days, and then they were put into this discrimination task. So the first part of this discrimination task is teaching the animals to touch the screen and then also to bar press to initiate the trials. And so the repeated mTBI models had similar days to touch criteria and to bar press, so they could learn the task just fine. But what we found was that the repeated mTBI animals had a significantly fewer correct responses, more incorrect responses, more correction trials, and significantly more trials to criteria. They were so impaired in this task, actually, that there was only 1 animal that actually passed the criteria. The trials were actually terminated 60 days after the initial discrimination trial. So we found that there's a significant cognitive deficit associated with repeated injuries. We also did some histology with these repeated injuries and found that indeed we do get an increase in GFAP on the repeated injury model, both in the cortex and in the corpus callosum. That's quantified here. And this is actually where one of the areas that we saw sex differences. So we do see some sex difference in the males here in the cortex on day 11. And then specifically a little bit earlier on in the females at day 5. And so we do see some sex-specific differences. But now we really think that we have a very nice model that we can of the repeated injuries that lead to long-term cognitive deficits. As I mentioned, we went out 60 days and the animals were still impaired. So we feel like this is a good model that we can now interrogate some of these underlying mechanisms. And so some of the things that we're looking forward to do is network dysfunction, how do the prefrontal cortex respond during this behavioral tasks. We have studies underlooking the structural deficits that are associated with these repeated injuries, and potentially a very nice preclinical model where we can look at different therapies or treatment in that early window to then hopefully prevent and encourage brain recovery and repair. So as I've kind of shown you without, I can't do this by myself. And I really want to give a big shout out to the Center for Brain Recovery and Repair, which is funded by NIGMS, our Department of Neurosciences, and the whole host of people that helped actually do the experiment. But more specifically, I'm lucky enough here to be in New Mexico, where we have a New Mexico Spreading Depolarization Consortium. So this is made up of both clinical and preclinical investigators really interested in looking at the role of SDs in a whole array of TBIs, ranging from severe to very mild, like I've shown you today. And so I want to thank everyone for participating. This is a great opportunity. I want to thank everyone for their time, and I'm happy to take any questions.
All right, thank you so much, Dr. Morton, for a fun and really engaging talk. First question is from Robert Dickinson, who says, really interesting presentation. I may have missed this, but at what time point after mTBI are the NSS impairments observed? Are these behavioral deficits observed concurrently with SDs/metabolic dysregulation? Is there any evidence of persistent NSS deficits? Have you looked at anxiety behavior at longer time points?
Great, thank you. That's a bunch of questions rolled in together. So sorry for not clarifying. The neurological severity scores are done immediately after. So that begins with our 10-minute open field task, like I showed you before. And then sequentially throughout. So then they're done very early on. So what I didn't show you today, which I probably should have, was we've done the neurological severity score at 3 hours, and all animals have recovered by that point. So we're looking at a roughly 2-hour window or an hour window where we have these behavioral deficits. As far as anxiety-like behavior long-term, we did do a zero maze task, but I think that was at 24 hours and we didn't find any deficits in that. Obviously there's more sensitive anxiety invoking things than that, but we haven't looked specifically at more longer term than just 24 hours. I hope
Next
that helped
question.
knock those off.
The next question is, thanks for an interesting presentation. In the mTBI model, did you find that the SD always occurred only once within minutes of injury? Were there any cases where there were recurring SDs following mTBI injury?
That is a very, very, very good question. And that's something we are very actively pursuing. So what I showed you pretty much all of today was where we detect a single SD from the impact in the anesthetized animal. And so as I've showed you with our rollerball experiments, and we have some 2P experiments as well, is that we're now moving that into awake and behaving. And so what we think is that when there's this reduction in cerebral blood flow, as I mentioned with those vicious cycle of SDs, we think that there's a potential when the brain activity increases when they recover from anesthesia, that there is potential for recurring SDs. We do have some very, very preliminary data to suggest that yes, there are recurring SDs, but it's very preliminary and we're still trying to tease that apart.
Okay, here's another question from David Brody. Thanks, Dr. Morton. This is an important line of investigation. What do you think would be the best approach to assess SDs in living humans after mTBI/concussion?
That's a great question. I mean, so as I mentioned with the clinical stuff early on with spreading depolarizations, is that you really need surface contact with the brain to be able to detect these. And so obviously that's not possible in the human running around a football field. But I think our best bet of identifying these in humans is FNIRS. So as some of you may be familiar with, FNIRS is a functional way of looking at cerebral blood flow. And I think that, you know, it seems feasible with the size of the newer instruments of those FNIRS that you could have one of those, say, on a sideline or something like that, where the sensors could be put onto the head relatively quickly. And if there are recurring SDs, I think that's our best bet of catching them. We're never going to catch, I shouldn't say never. I don't think we're going to catch the first one, right? Because there's so much going on. But I think if there are recurring SDs, we'll be able to detect them with these and maybe even more sophisticated EEG. But I think FNIRS is our best bet.
We have a question from Alyssa Geen who says, this may be naive, but do you believe that a single mTBI impact in humans is always recoverable?
I do. Yeah, I think, so those of you who may or may not be familiar with spreading depolarizations, they've been tightly linked to migraine aura. And so this is first described by how the migraine aura kind of propagates across the visual field. That's been linked directly to the propagation of the SD. And there's actually been some fMRI studies directly showing that the propagating wave is associated with the migraine aura. So in that case, people are completely healthy, their brains are healthy, their cerebral blood flow is normal, everything is good, the SD goes through, and so they recover just fine. And so I think a single SD in a healthy brain, I think, is absolutely recoverable. When it becomes problematic is when there's recurring SDs or the brain is compromised in some other way. And so I think that's a really great question. I think that I think spreading depolarizations may be more prevalent than we really think, just because we can't detect them. But it's a very great question.
And David Brody came back with another question. Do the animals have migraine-like features such as photophobia, phonophobia, allodynia, like experience with like actual migraine patients?
That's a great question. Thank you for asking. We've actually, some of our most recent studies are looking directly at that. We've actually have a collaboration that just got funded through the DoD with Casey Brennan in Utah, who's a migraine researcher. And we're doing just that. We're looking to see if there's photophobia, photosensitivity, allodynia, things like that. There was one recent study that just came out of Cenk Ayata's lab that showed that the optogenetic stimulation of the SD was associated with orbital sensitivity. So we think there may be a link between spreading depolarizations and headaches specifically, more general headaches. But that link is still not fully fleshed out. But I think it's a great question. I think at least with the movement in the home cage and things like that, there's definitely indications of headache-like behavior, and we're just beginning to tease all that out right now. So great question. Thank you.
And I guess Shambag says, thank you, Dr. Morton. Have you explored perivascular CSF dynamics as a part of the glymphatic system in your model?
That's a great question as well. We would love to. As I mentioned, we're just now starting some multi-photon imaging, looking at cerebral blood flow, and that would be one of the things we would love to look at. And again, I'm always open for collaborations. If people have expertise doing such things, we would love to look at perivascular things just like that. Exactly. Thank you.
Rosie Paterno says, really interesting talk. Do you see any difference in the spreading depolarization events and frequency or duration after multiple impacts, and if they correlate with more severe phenotypes?
Again, a really great question. We're looking at that. So we have our repeated model, which occurs every 24 hours. We're looking at that right now. But at this point, very early on preliminary studies, it doesn't appear so. But we also think that if we look at our cerebral blood flow data that I showed today, it looks like things are relatively recovered within that 24-hour period. But then again, I showed you at the end of the talk, we do have cognitive deficits that persist after the 4 injuries. So there's something going on. If it's directly linked to the SD or not is completely up for discussion, but we're really looking into that. But our initial studies don't indicate that within the 24-hour window, at least, the spreading depolarizations properties don't seem to be different at this point.
Another question is, did you find any age differences in your rodent model with SDs or the behavioral data?
That would be a great question. We've really limited our studies to focus in on that 8 to 12 week time point. But it would be incredibly interesting to go in both directions, right? To look at more of an adolescent age and an older adult, I think would be really important. I think it'd be really interesting to find where, what we do know, at least from the clinic, is that, often these simple falls and things like that can lead to subdural hematomas and serious injuries, right? And so it would be very interesting to expand our ages in both directions, but we haven't had the opportunity to do that quite yet.
Okay, Dr. Morton, we're on our last question. So
Okay.
Great talk that we can do for today. Great talk based on the pathophysiology of SD you described. Where do you think is the most promising phase is for potential therapeutic?
So I think the best bet for therapeutics, I think, is still within the first few days. And how that relates to spreading depolarizations is kind of yet to be determined, right? So I showed in one of the slides, we hypothesize that recurring SDs may contribute to the extended period of cerebral blood flow deficits and things like that. Of course, we don't have the data yet to prove any of that, but I still think that it's the first few days. I think we're going to have the best bet on any therapeutics, even if it's involving SDs or not. I think that's really a critical window into the injuries, and if we can promote brain recovery during that early phase, I think it's our best bet. And that may not have anything to do with spreading depolarizations, but I think it's a time point that's probably most critical.